So, hello students, welcome to once again to my YouTube channel, and in succession to the physiology lectures what I have been taking up these days. The male reproductive system, I have already discussed the functional anatomy of the testes and the parts of the sex organs. So in succession to that video, this is my third video where I'll be mainly talking of the spermatogenesis and the functions of the Sertoli cells. So students, beginning with just a one-line brief definition, one-line brief introduction as to how in what manner today I'll be talking of the histology as to how the spermatogenesis is going to take place and the site where it is occurring. So in the previous video, as I've already said, that the spermatogenesis is going to occur in the seminiferous tubules. Particularly, it is the seminiferous tubules what are lying in the pyramidal lobes of the testis. The spermatogenesis is going to occur, and the spermatogonia are the cells, are the primitive precursor cells what are lining the seminiferous tubules, which ultimately undergo various stages, different stages, to give rise to the mature spermatozoa. So today's video, I'll be talking of the histological section as to how the seminiferous tubules looks within. So this is the diagram what I have already drawn, and this diagram is representing that the testis is going to contain 200 to 300. These are the pyramidal lobules what I have drawn. And these pyramidal lobules are the sites where the seminiferous tubules are present. So in each testis, approximately 200 to 900 seminiferous tubules are present, 400 to 600 seminiferous tubules, they are present, and within each pyramidal lobule, as such, there are 1 to 4 seminiferous tubules. So these seminiferous tubules, these are the sites where the spermatogonic cells, which are the precursors of the mature spermatozoa, are present. And these seminiferous tubules, they are being lined by the spermatogenic cells. So on looking at the histological or the cut section through the seminiferous tubules, it has been seen that this seminiferous tubules is basically consisting of 3 layers. That is, the wall of the seminiferous tubule is consisting of 3 layers. What is observed under a microscope, what we can see that the outer capsule of the seminiferous tubule, it is called as the tunica propria. So the outer capsule of the seminiferous tubule, it is the tunica propria what is present, and just beneath it, that is in the middle, there is going to lie a thin big homogeneous basement membrane. So this is the homogeneous basement membrane what I have drawn. So outer the seminiferous tubule, when looked under a cross section or in a cut section, the histology, the pattern in which the cells, the contents are lying, outermost, it is the outer capsule or the tunica propria. Within it, it is the thin homogeneous basement membrane is present, what I have drawn. And this thin homogeneous basement membrane is the structure on which the spermatogonic cells are going to lie. So this thin homogeneous basement membrane, what is present, it is chiefly consisting of 2 types of cells. Firstly, there are presence of the spermatogenic cells or the germ cells. So these are the spermatogenic cells what I have drawn, and these spermatogenic or the germ cells, these are lying on the basement membrane. And in addition to them, these spermatogenic cells, these spermatogenic cells, they are containing in between them the Sertoli cells. So actually, these spermatogenic cells or the spermatogonic cells, what are present, they need nourishment. Actually, the mature spermatozoa, what is being formed, so these spermatogenic cells, they actually require the constant supply of nourishment and nutrients. So it is the Sertoli cells, which are tall columnar cells and which have cytoplasmic connections to the spermatogenic cells to provide nutrients and nourishment to the developing spermatogenic cells. All right, so these are the Within the seminiferous tubules, particularly the two types of cells are present. Firstly, it is the spermatogenic cells, which are going to lie in different layers. Different 3-4 layers of the spermatogenic cells are there, and between these spermatogenic cells, tall columnar cells what are present, Sertoli cells, and these Sertoli cells have cytoplasmic connections through which the spermatogenic cells receive nutrients and nourishment for the maturation into the spermatozoa. So it is the brief histological sections, and as we can already say that since in children the testis is not fully developed, the spermatogenic cells are in the primitive stage, and this primitive stage of the spermatogenic cells, it is called as the spermatogonia. So spermatogonia, it is the precursor or the primitive cells which is ultimately going to form the mature sperm, and with the onset of puberty, the spermatogonia develop into sperms through different stages. So this video I'll be mainly talking of the different stages of the spermatogenic cells. So the first stage what is seen, it is the stage of proliferation. So the stage of proliferation, we can find that these spermatogenic cells is during this stage that the rapid proliferation of the spermatogenic cells occur, and these spermatogenic cells contain the diploid number of chromosomes. So the first stage, that is the stage of proliferation, as we can see, during this stage, there is rapid proliferation of the spermatogenic cells or the spermatogonia. And in this, the chromosome number is diploid. So there are basically 23 pairs of chromosomes, out of each pair, one is from the maternal side and one is from the paternal side. And these 23 pairs of chromosome, they continuously undergo mitotic divisions to produce different to produce a large number they divide continuously up till 7 generations of spermatogonia, and ultimately they will reach the next stage, that is the stage of growth. So during the stage of proliferation, it is the spermatogonia which undergo continuous rapid mitotic divisions, and this rapid mitotic divisions of the spermatogonia is going to continue till the 7 stages. There are usually the 7 generations of spermatogonia are present, and during the last generation, the stage of the spermatogonia, they enter the stage of growth, and this after the stage of growth, they will ultimately migrate, they will ultimately transform into the primary spermatocyte. So this spermatogonia during the stage of proliferation rapidly undergoes mitotic divisions. These mitotic divisions are going to continue up till 7 generations, and after the 7th generation is the last generation. This will be also containing the cytoplasmic connections from the Sertoli cells to receive the nourishment, and during this stage of proliferation, the spermatogonia migrate from the base or from the basement membrane towards the lumen of the seminiferous tubules. So the next stage, it is the stage of growth. Now the last stage of the spermatogonia is going to transform into the primary spermatocyte. So during this stage, the primary spermatocyte grows into a large cell. Apart from growth, there is no other change in the spermatocyte during this stage. So primary spermatocyte is that stage. After entering this stage, the only growth is going to occur within the primary spermatocyte. So during the stage of growth, the primary spermatocyte, which is also containing the diploid number of chromosome, and they also will the cells the spermatogenic cells which have transformed into the primary spermatocyte during the stage of growth, they will show a growth in size and nothing else will be seen. The diploid number of chromosomes will be present in the primary spermatocyte as well. Now, next, it is the stage of maturation. So, it is during the stage of maturation that there are going to occur the reduction division. So, the primary spermatocyte, which was containing the diploid number of chromosomes, will transform into the secondary spermatocyte, and it is the transformation from the primary to the secondary spermatocyte that the reduction division or the meiosis is going to take place. So the meiosis will occur when the primary spermatocyte will transform into the secondary spermatocyte, and this stage of maturation, that is during the third stage, this stage of maturation is basically divided into two stages. That is the first stage, that is in the case of first meiotic division, the primary spermatocyte undergoes the reduction division to give rise to secondary spermatocyte, and during the stage 2, the secondary spermatocyte further undergoes reduction division to form two spermatids. So this was the stage of maturation, and finally we have the stage of transformation where the secondary spermatocyte is going to transform into the spermatid. So the fourth stage, it is the stage of maturation, transformation, where the secondary spermatocyte will transform into a mature spermatozoa. Okay, and finally, by means of spermiogenesis, this transformation from secondary spermatocyte to from spermatid to matured spermatozoa, the process is known as spermiation, the spermiogenesis, and they are released into the lumen of the seminiferous tubules through the process which is known as spermiation. So spermiation is that process by which the mature spermatozoa are released into the lumen of the seminiferous tubule, whereas the transformation of the spermatid into the matured spermatozoa is known as spermiogenesis. So students, this was a brief discussion about the various stages through which the spermatogonic cells they undergo, through which to give rise to the mature spermatozoa. So just as already I've said that within the between the between the spermatogonic cells, they are going to lie the Sertoli cells. So these Sertoli cells, they are actually the nurse cells or the cells which are going to provide nutrition to the developing spermatogonia. So these Sertoli cells, they have one basic function, that is they are going to provide nourishment to the developing spermatogonia. And these Sertoli cells, as I've already said, that between the spermatogonic cells and the Sertoli cells, there is going to occur cytoplasmic connections. And it is through these cytoplasmic connections that the spermatogonia are going to receive the nutrition. So until and unless the mature spermatozoa is being released into the seminiferous tubules, these cytoplasmic connections between the spermatogonia and the Sertoli cells is maintained. So a brief discussion, brief functions. What are the functions of the Sertoli cells? So when looking at the functions, as I've already said, the first function is to provide nourishment to the developing spermatogonia. And is after that, the next function, what we can see, it is that they Sertoli cells, they are chiefly responsible for the production of an enzyme which is known as aromatase. And this aromatase enzyme is chiefly responsible for the conversion of androgen to estrogen. So this aromatase enzyme is being produced by the Sertoli cells. In addition to it, the third function, what can be looked upon, it is the androgen binding proteins. So these are the androgen binding proteins which are very essential for the testosterone activity, particularly during the process of spermatogenesis. So students, it is the androgen binding proteins. These are being produced by the Sertoli cells, and this androgen binding protein is very essential for the testosterone activity during the process of spermatogenesis. So the second function, the third function I've discussed. Similarly, the fourth function, what can be looked upon as, it is the estrogen binding proteins. So this Sertoli cells, they are responsible for the production of the estrogen binding proteins. And fourth or the fifth function, what we can see, that they secrete inhibin. So as we can already see that it is finally the testosterone gets modified into the estrogen, and this estrogen has a negative feedback mechanism on the pituitary to lesser the secretion of FSH and LH. So particularly this Sertoli cells, they secrete inhibin, which is going to inhibit the follicle stimulating hormone released from the anterior pituitary. So until and unless the follicle stimulating hormone is not being secreted, the spermatogenic cells, they will not be getting activated, their transformation will not begin. So firstly, these Sertoli cells, they are going to secrete the hormone inhibin, which is going to inhibit the follicle stimulating hormone release from the anterior pituitary. And finally, the last but not the least, and the very, very important function of the Sertoli cells, that they are releasing the Mullerian inhibiting substance or the Mullerian regression factor. So students, as you all might be very well aware, that it is actually the Wolffian duct Wolffian duct, which is responsible for the production of the accessory sex organs in male, and it is the Mullerian duct which is responsible for the production of the accessory sex organs in case of females. So actually, when these Sertoli cells, they release the Mullerian regression factor, so they are going to, in a fetal testis, when this Mullerian regression factor or the Mullerian inhibiting substance is released from the Sertoli cells, the regression of the Mullerian duct is going to occur, and with the regression of the Mullerian duct, the fetus will not going to transform into a female fetus, and that will lead to the development of the accessory sexual male characteristics. So secrete the it is the Sertoli cells which are chiefly responsible for the secretion of the Mullerian regression factor in fetal testis. And ultimately, the degeneration of the Mullerian duct will occur, and the Wolffian duct is going to produce the male genital system. The Wolffian duct will give rise to the male accessory sex organs. So the most important function of the Sertoli cells, it is the secretion of the Mullerian inhibiting substance, and which is responsible for the regression of the during the differentiation of fetus. So students, do remember a very, very important fact, that it is the testosterone which is responsible for the inhibition of the Mullerian duct, for the regression of the Mullerian duct. And in addition to the testosterone, it is the Sertoli cells which are going to release the Mullerian regression factor, which is ultimately going to regress the Mullerian duct, leading to the development of the male accessory sex organs through the Wolffian duct. So students, this was a short discussion about the different stages of spermatogonia and the process as to how the sperm gets matured, and the different functions of the Sertoli cells. So students, if you do like this video, give this video a thumbs up, and if you do have any queries or comments, and if you have any topics what you want to be discussed by me, you're most welcome to comment me in the comment section. Thank you for watching.